Hello everyone and welcome to my channel, and in this lecture we are going to study about the sterilization techniques. The sterilization technique is a very important technique in a different kinds of industries. For example, the pharmaceutical industries, dairy industries, food industries, also in different kind of labs including a microbiology lab, and also in a hospital there is a need of a proper sterilization. The definition of sterilization: sterilization is defined as a process where all the living microorganisms, including bacterial spores, are killed. In this technique, vegetative cell as well as bacterial spores are killed. Sterilization can be achieved by a different way, by physical means, chemical means, or a physico-chemical means. And the chemical that are used as a sterilizing agent are called as a chemisterilant. The sterilant is nothing but a chemical agent that is used for a sterilization purpose. Disinfection. The disinfection is a process of elimination of most pathogenic microorganisms, but in this disinfection here is not a killing of bacterial spores, where it is only killing of vegetative cells only, and it can be applied on an inanimate object. And this disinfection was first time given by a scientist, Joseph Lister. The next point, it is about the decontamination, and decontamination is the process of a removal of a contaminating pathogens from an article by a process of sterilization or a disinfection. It is the use of physical or chemical means to remove or inactivate or to destroy a living organism on a surface so that the organisms are no longer infectious. Sanitization: this is the process of a chemical or mechanical cleansing, and it is applicable in public health system. Usually it is used by a food industry. It reduces microbes on eating utensils to safe. Asepsis: it is a employment of a technique to achieve a microbes free environment. It is a employment of a technique to achieve a microbe free environment, such as use of gloves, use of air filters, use of UV ray. The use of UV ray, it can be applicable in a laminar airflow where there is a maintenance of a strict aseptic condition. Also, during microbial techniques, there is a use of a aseptic condition for inoculation of organism onto a culture media, where there is a use of a microbe free environment. Next definition, it is of antisepsis. In antisepsis, there is a use of chemicals, and that chemicals are called as antiseptic. And these antiseptics are used to make skin or mucus devoid of pathogenic microorganisms. The next term, it is a bacteriostasis. Bacteriostasis is a condition where we can inhibit the growth of a multiplying bacteria. But in this bacteriostasis, we does not kill the any bacteria, but here we just inhibit the growth of bacteria. Next point, bactericidal. It is a chemical that kills or that inactivates a bacteria, and such chemicals are called as bactericidal chemicals. They may be bactericidal, virucidal, fungicidal, microbicidal, sporicidal, and tuberculocidal or germicidal. Means the cidal name or a cidal suffix shows that it is used for a killing purpose of a particular kind of organism. Antibiotics. The antibiotic substance are the substance produced by one organism, and this substance inhibit or kills the another microorganism, and such substance are called as a antibiotic. The example of antibiotic, we know that the first discovered antibiotic, that was a penicillin. It is discovered by a scientist, Sir Alexander Fleming, and the producing organism for this antibiotic was a Penicillium notatum, and this antibiotic produced by this Penicillium notatum, it is one fungi, and this acts on the gram-positive bacterial pathogens. Antibiotics are the substance produced by one organism that inhibit or that kills another microorganism. The next term, it is a thermal date point. Thermal date point, it is the temperature, or it is the lowest temperature at which all cells in a culture killed within a 10 minutes. Thermal date point, TDP, is nothing but a lowest temperature at which all the cells in a culture get killed in a 10 minutes. Next point, thermal date time, TDT, it is a time during all the cells in a culture get killed. Now, very important about the highly resistant organism to the disinfectant. The highly resistant organism or the microorganism is prions. The prions are nothing but a infectious protein material, and this infectious protein material is causing a number of diseases to a plant. The one famous example of a prion, that is a Creutzfeldt Jakob disease causing prion, that is a CJD, and this disease is because of a prion. Also the mad cow disease and a Kuru disease, these examples are of prion diseases, and the prions are nothing but a infectious protein material, and these are hardest to kill by any disinfectant. Then next, spores of a Clostridium are hardest to kill mycobacterial tuberculosis organisms. Then next, non-enveloped viruses. Then next, the fungi, for example Candida. Then next, vegetative bacteria, and then next, enveloped viruses. If we come from top to bottom, at the top we can see for a hardest organism, and these are resistant to a number of disinfectant. Now, the physical methods of sterilization. In a physical method, we can make a use of a heat, and this heat is considered as a most reliable method for a sterilization. But by using heat, we can sterile the material which can withstand for a heat. By using heat, we cannot sterile a heat sensitive material, for example plastic or a plastic like wares. Now the next point, it is about the action of heat, in which manner or how the heat can inhibit or heat can kill the growth of organisms. The dry heat acts by a protein denaturation and toxic effect of elevated levels of electrolytes. The most moist heat acts by a coagulation and denaturation of a protein. We can make a use of dry heat or a moist heat, but the moist is a superior to a dry heat in action, because the moist heat has a high penetration power. So moist heat is a superior for the sterilization purpose. Dry heat can be used like a following way: red heat or a flaming. For inoculation purpose, we make a use of a transfer needle, or we make a use of a bacteriological loop or a nichrome wire loop. Before inoculation, the such utensils like nichrome wire loop or a transfer needle should be sterile, and for this sterilization we can make a use of this technique, that means red heat or a flaming. And also the forceps and a spatula are also sterilized by using a flaming. In this technique we just have to simply hold these utensils into a Bunsen flame till they become red hot. After a red hot, these wares or these utensils get sterilized. The next, hot air oven, and using of a hot air oven for a sterilization process, it was introduced by a scientist Louis Pasteur. The material to be sterilized by using hot air oven is a glasswares like pipettes, petri plates, and test tubes. And here there is use of a temperature 160 degrees Celsius temperature for 1 hour. There are a different time and temperature relationship for holding 60 minutes at 160 degrees Celsius temperature, 40 minutes at 170 degrees Celsius temperature, and 20 minutes at 180 degrees Celsius temperature. These are the time temperature relationship and holding time. Then insertion, in insertion, here we destroy a contaminated material by burning them into an incinerator. Here there is one incinerator. Here the hospital waste or biological waste, such kind of waste are burned, and so that they get the organism present in that material get killed, and also the material also get killed. The article or a material like dressings in a hospital, animal carcasses, pathological material, beddings, clothes of patient are subjected to such kind of sterilization. Here the second image is of a oven, and the first image, this image is a sterilization of a transfer needle. Here we can see for a red hot of a wire loop. Sterilization by a moist heat. The moist heat acts by a coagulation and denaturation of protein. Also, by using moist heat, the activity of enzymes get weakened. Moist heat can be used at a 3 different temperature: below 100, at 100, and more than 100. Below 100, the pasteurization. In a pasteurization, there is use of a temperature that is a 72 degrees Celsius for 15 minutes or half an hour, not 15, half an hour. And this process of pasteurization was given by a scientist Louis Pasteur. This process of pasteurization nowadays used in a food and dairy industry, and this is mainly used for disinfection of milk. There are two methods of pasteurization. One is a holder method and another one is a flash method. In a holder method, the milk is kept for 30 minutes for 63 degrees Celsius temperature, whereas in a flash method, milk is kept for 15 seconds for 72 degrees Celsius temperature, and after this each and every particle is cooled down and packed into a sterile plastic bag. This is a process of pasteurization, and this is one of the sterilization process in which there is use of moist heat. At a temperature 100 degrees Celsius, boiling of water at a 100 degrees Celsius temperature kills most vegetative bacteria and virus. Certain bacterial toxins such as staphylococcal enterotoxins are also heat resistant. Some bacterial spores are resistant to a boiling and survives. Hence, this is not a substitute for a sterilization. At a 100 degrees Celsius temperature, we cannot sure that all the endospores or a bacterial endospore get killed. For this purpose, the temperature more than 100 have to use. So this is not a perfect sterilization, and also the toxins produced by a number of bacteria are heat resistant and does not get inactivated at a 100 degrees Celsius temperature. So for this we have to use a temperature more than 100 degrees Celsius. At a temperature above 100 degrees Celsius, the equipment, that is a autoclave. In this autoclave, moist heat is used, and there is a temperature more than 100, that is a 121 degrees Celsius temperature. At this temperature, there is a pressure of a 15 Pascal, so that because of such pressure, the articles or the material get sterile, all the endospore get killed. The next point, it is of radiation. Two types of radiation can be used: ionizing and non-ionizing. The non-ionizing rays having a low energy and low penetration power, whereas ionizing rays are having a high energy rays, and these rays having a good penetration power. So basically the ionizing radiations are used for a sterilization purpose. During a radiation, the radiation does not generate any kind of heat, so it does not increase a temperature during a process, so it is called as a cold sterilization. Cold sterilization means a use of radiation for a sterilization. Two type of rays can be used. One is a non-ionizing and another one is a ionizing. Non-ionizing rays, the rays of a wavelength longer than a visible light, are non-ionizing. Microbicidal wavelength of a UV rays lies in a range of 200 to 280 nanometers. This is a range of a wavelength of a UV rays which is a microbicidal, 200 to 280 nanometers. But at 260 nanometers, it is a most effective, because at this wavelength the DNA of an organism accept a more amount of rays. So because of this absorption, the DNA get denatured, it get mutated and organism get killed. So this is a best wavelength for a killing purpose of the organisms. Ionizing rays, the ionizing rays are of two types: particulate and electromagnetic rays. The high speed electrons are produced by a linear accelerator from a heated cathodes, and electron beams are employed to sterilize the article like syringe, gloves, dressing packs, foods, pharmaceutical products. And in this, or by this using ionizing rays, the sterilization is carried out within a few seconds. The electromagnetic rays such as gamma rays, they can emanate from a nuclear disintegration of a certain radioactive isotope like cobalt 60, and because of this electromagnetic rays or a gamma rays, organism get killed. They have more penetration power than electron beams but requires a longer time of exposure. Filtration, a number of filters are used. The diatomaceous earth filter or membrane filters. But in a filtration there is no any killing of microbes, but the material can be separated out of them. Membrane filters with a pore size between 0.2 to 0.45 micrometers are commonly used to remove a particles from a solution. The heat sensitive material or a liquid material can be sterilized by using a filtration. The heat labile liquids such as serum, antibiotic solution, sugar solution, vitamin solution, urea solution, these liquid solutions are sterilized by using a filtration process. The next point, a chemical method of a disinfection. In this method there is use of a chemical for a disinfection, and the chemical that destroy a pathogenic bacteria are used for the purpose of sterilization. Alcohols. The alcohols like ethyl alcohol, isopropyl alcohol and a methyl alcohol are used for a sterilization purpose. The mode of action of alcohol is alcohol can dehydrate the cell and they disturb a membrane, because in a pathogen their cell wall contains a lipid. The lipid of a pathogen present in a cell membrane get swallowed or soluble into a alcohol, so that lipid get leaked from a cell membrane and the organism get killed. Means the alcohol disturbs a membrane of a pathogen and also it causes a coagulation of a protein. The aldehydes. The aldehyde, number of aldehydes can be used, formaldehyde and butylaldehyde. These aldehydes can carry out a alkylation of amino, carboxyl or hydroxyl groups, and so they damages a nucleic acid and kills a microorganism. Also they kills a pores. Phenol. The use of phenol as a disinfection was given by a scientist Joseph Lister, and this was the first disinfection agent. And also by using a standard phenol we can estimate a phenol coefficient of other chemical disinfection agent. Phenol coefficient means phenol has two advantages. One, it is a chemical disinfection as well as other chemical disinfectant agent are compared to the phenol. Mode of action, it acts by a disruption of membrane, precipitation of protein and inactivation of enzyme. The next chemical is a hydrogen peroxide, and this hydrogen peroxide is a strong oxidizing agent and it oxidizes the microorganism so that the organism get killed. The hydrogen peroxide releases a nascent oxygen. This nascent oxygen damages a proteins and a DNA, so the organism get killed. The dye, the acridine dye, for example acridine dye, the acridine dye are a bactericidal because the acridine dye interacts with a nucleic acid and nucleic acid get mutated, hence the organisms get killed. Example, aniline dyes such as crystal violet, magenta green and brilliant green. Here are some examples of biological monitors. The sterilization by a steam. For this process the monitor or a indicator is a Geobacillus stereothermophilus. To check out a sterility testing by a steam, we have to check that whether this organism Geobacillus stereothermophilus is there, or if we inoculate a pores of this organism and if these pores are grow on a material which was sterilized by a steam, then we can say that that material is not properly sterile. Here it means that for a sterility testing by a steam, this organism completely free from that material, If there is a presence of geobacillus stereothermophilus on a material which were sterilized by a sting, then there is no surety of sterility. Means the material is faired for a sterility testing. For a dry heat, there is use of pores of Bacillus atrophes. Formerly, it was a Bacillus subtilis. For a sterilization of ethylene oxide or a ETO, there is a monitor, that is a Bacillus atrophes. Thank you.